Meiosis is a process of nuclear division that reduces the number of chromosomes in new cells to half the number in the original cell. The purpose of meiosis is to produce gametes, which are haploid reproductive cells. Human gametes are sperm cells and egg cells. Sperm and egg cells contain 23 chromosomes. The fusion of a sperm and egg cell results in a zygote that contains 46 chromosomes.
Phases of meiosis 1 include interphase, prophase 1, metaphase 1, anaphase 1, and telophase 1. And the phases of meiosis 2 proceed with prophase 2, metaphase 2, anaphase 2, and telophase 2.
This is interphase. During interphase, the cell is not undergoing meiosis or dividing. Interphase consists of regular activity, and when it prepares to divide, it undergoes the G1 synthesis and G2 phases of interphase.
In G1, the cell's contents are duplicated. In synthesis, each of the 46 chromosomes are duplicated. And in G2, the cell double checks the duplicated chromosomes for error, making any repairs.
Meiosis 1 officially begins with prophase 1.
Prophase 1. DNA coils tightly into chromosomes and spindle fibers appear. The nucleolus and nuclear membrane disassemble. Every chromosome begins to line up with its homologue. This pairing of homologous chromosomes is called synapsis, and each pair of the homologous chromosomes are called a tetrad. In each tetrad, chromatids of the homologous chromosomes are aligned lengthwise. This is so the genes on one chromosome are adjacent to the corresponding genes on the other chromosome. During synapsis, the chromatids within a homologous pair twist. Portions of the chromatids may break off and then attach to adjacent chromatids on the homologous chromosome. This is known as crossing over, and it permits the exchange of genetic material between maternal and paternal chromosomes. Genetic recombination starts because of this.
The next phase of meiosis 1 is metaphase 1.
Metaphase 1. The tetrads line up randomly along the midline of the dividing cell. The location of each pair of chromosomes is random due to the poles of the cell. Spindle fibers from one of the poles attaches to the centromere of one homologous chromosome, and spindle fibers from the other pole attach to the other homologous chromosome of the pair.
Next is anaphase 1. In an anaphase, each homologous chromosome moves to an opposite pole of the dividing cell. The separation of the homologous chromosome is called independent assortment, and this results in genetic variation.
The last phase of meiosis 1 is telophase 1. In telophase 1, the chromosomes reach the opposite ends of the cells and cytokinesis begins. The cells contain a haploid number of chromosomes.
The original cell produces two new cells, each containing one chromosome from each homologous pair. The new cells contain half the number of the chromosomes of the original cell. However, each new cell contains two new copies as chromatids because the original cell copied its DNA before meiosis 1.
After telophase, meiosis 2 officially begins with prophase 2. In prophase 2, spindle fibers form and begin to move the chromosomes toward the middle of the dividing cell. The metaphase plate begins to form as well.
Next is metaphase 2, and here the chromosomes move to the midline of the dividing cell, and each of the chromatids will be facing opposite poles of the dividing cell.
Next is anaphase 2. In anaphase 2, the chromatids separate and move toward opposite poles of the cell.
The last stage of meiosis is telophase 2. In telophase 2, a nuclear membrane forms around the chromosome in each of the four new cells. Cytokinesis 2 occurs, resulting in 4 new haploid cells. Each of these new cells contains half the number of the original cell's chromosome. And this concludes the phases of meiosis. Thank you.